Professor,
Yes.
this is Ariana Watson, and we also have a test on Wednesday as
Yeah, that's right. Yeah, so we might as well start and just let me comment on that. Today you have quiz 6, and on Wednesday you'll have test 2, which will cover everything since the last test, since test 1. And I'll send you an announcement laying out exactly the topics that have been covered since test 1. So you'll have a record of that. But for your studying purposes, if you just go back to the end of what was covered on test 1, everything from there on, except today's discussion will not be on test 2. Well, since you're having a quiz today,
Good afternoon.
our
I have a question about the today's test. It includes coronavirus. We don't have any enough information about the uh, slides or in the textbook about the coronavirus vaccine. How should
yeah,
we?
you have slides. You have
Oh,
slides on coronavirus, and it includes a discussion of vaccines. So you can find that either in the slide set or listen to the recording. Okay, let's start. I'll shorten today's class period by 15 minutes because that's what you will need for your quiz. This is the topic for today. Cell division, mitosis, and meiosis. These two processes occur only during cell division. In order for organisms to grow, develop, reproduce, repair themselves, cells have to divide. And these two processes, mitosis and meiosis, ensure that genes are transferred accurately from one generation to the next. So let's start with a look at karyotypes. A karyotype is a visual display of chromosomes. So that means you view a karyotype with a microscope. Now in humans, a haploid cell, a copy number, chromosome number, is 23. And that means 22 autosomes and one sex chromosome. The diploid chromosome number is just twice that, 46, with 44 autosomes and 2 sex chromosomes. So you see in this, these two pictures, a human female and a human male. If you look at each of those chromosomes, they're identical in their shapes between males and females until you get down here to the bottom right. In a female, you have 2 X chromosomes. In the male, you have 1 X, which is just like a female X. But the second is a Y. It's a very small chromosome. That's what determines the difference between a male and a female. So we're all, to some extent, females because we all have at least 1 X, but we males also have a Y. Here is another picture of karyotypes. The one on the left is sort of blurry, but it doesn't matter. What I'd like you to do is look at these chromosomes. They're very pretty. They're stained, that's why they have different colors. and see if you can tell which one is male and which one is female.
This is Zach. I would say the left is male and the right is female.
Yeah, you're right, because you spotted in the female there are 2 X's, and the male there's 1 X, and then that little blob there is a Y. Now, some disorders in humans are caused by abnormal karyotypes. So here we have 2 side by side, representing 2 different abnormalities. Take a look at those and tell me what's different. What's the aberration in the one on the left?
The X and Y are swapped.
Well, X and Y are both there. It doesn't matter which side they're on, as long as they're there. There's something else that's different about.
So there is missing in Y, Agaric speaking.
Say that again.
There we are missing Y chromosome in the right side, but in the left side we have Y and X, so it's male. And
Yeah.
in the female we're missing another X.
Yeah, okay, that's correct. That's the abnormality in the female. What's the abnormality in the male?
Oh,
Bill
there's
Steinman
one more, the
speaking.
21,
Yeah.
right?
There's 3 copies of 21. Yeah, that's right. So let's go a little further on this and look at these aberrations. So more about chromosomal aberrations. We have the Down syndrome and the Turner syndrome. The Down syndrome is 3 copies of chromosome 21. And this can happen in either males or females. And these trisomies are caused by what's known as non-disjunction. It's a word you should remember. It means that chromosomes, when cells divide, the chromosomes don't separate properly. And that's called non-disjunction, and that can occur in either the sperm or the egg. You've all seen people with Down syndrome, so you know what most of the symptoms are. I won't go through them all, but they're quite distinctive. But people who have a trisomy 21 usually live relatively normal lives and quite long lives, 50 years or more. Now, the Turner syndrome, you've already noticed that Turners are missing an X. They have 1 X and no Y, so they're female. And people with this have delayed development. They're short in stature, never have puberty, they're infertile, have abnormal hearts, and usually they have relatively short lives. This is a picture of a girl with Turner syndrome. Now, every chromosome has the possibility of a trisomy. That is 3 copies instead of 2. Most of them are lethal. They abort during pregnancy. I've listed here the ones that can actually be born. Trisomies at 18, 13, 8, 22, and 9. They can survive through birth, but they live a very short time, anywhere from a year or less to a matter of a few days. Now, trisomy X, 3 copies of the X chromosome, is different in that girls who get that develop and live relatively normally. There are other chromosomal abnormalities. Males that get 2 X's and a Y, they are male, but They have a certain number of female characteristics and a low testosterone, the male hormone. Males can also have two Ys, but as long as they have an X, they're okay, and they live mostly normal lives. There's some reports that males with two Ys are more aggressive than those with one Y, but that isn't firmly established. All right, now let's get into cell division and the two main processes, starting with mitosis. So what are the consequences of mitosis? You start with one cell, and that cell can be either haploid or diploid. But it duplicates its DNA, and you remember that process, the 6 steps in DNA replication. And then it divides. So the result is 2 cells that are identical to their progenitor. One cell just becomes 2, and there's no genetic variation introduced. One of the keys to mitosis is just that. You get an exact copy of the starting material. So this slide we'll spend a little time on because you need to remember most everything that's on this slide. So let's start with the various different phases of mitosis. And if you want to watch a video, the URL for the video is given here. So at interphase, the cells appear inactive. They don't look like they're about to do anything, but the DNA has already replicated in interphase. The 6 steps of DNA replication have occurred. And there's the centriole divides and proteins are produced. Now in prophase, the nucleolus phase fades. We'll come back to the nucleolus in a bit. And chromatin, that too, condenses into chromosomes, each of which comprises 2 identical chromatids. So each chromosome is double. Now in prometaphase, over here, the nuclear membrane breaks down, the nucleus disappears, and the sister chromatids are joined by centromeres. Talk about that in a bit. Each centromere has proteins called kinetochores, and the function of the kinetochores is that's where the spindle fibers attach. And as you'll see, the spindle fibers are important in the separation of the chromosomes. A metaphase is where all the chromosomes line up in one plane at the center of the cell. It's a very distinctive configuration of the chromosomes. And then at anaphase, these spindle fibers, which are attached to the centromeres of each chromosome, they shorten. And in the process of shortening, they pull the chromosomes to opposite poles. So half the chromosomes go to one side of the cell, the other half go to the other. Then in telophase, the chromosomes arrive at the poles and the spindle fibers disappear, they're not needed anymore. And then finally the cell has to divide, and that's called cytokinesis, or a contractile ring. I'll show you that in the next slide. In animal cells, a contractile ring forms and cleaves the cell into 2 daughters. Now, I'd like to focus on this. Let's see. On the upper right here, which is the anatomy of the nucleus in interphase. You'll see the things that you're familiar with already, like the nuclear envelope, nuclear pores. And the chromatin, and what is chromatin? Well, it's long, relaxed chromosomes. So before an interphase, this is before the chromosomes start to condense, they're still chromosomes. They're long chains of DNA, but they're relatively relaxed and very long. Now, what is the nucleolus? I'd indicated that in prophase, the nucleolus disappears. Well, it's an organelle within an organelle, within the nucleus. It's composed of ribosomal RNA and protein, and its function is to produce ribosomes. These ribosomes are then exported through these nuclear pores into the cytoplasm, where they join the membrane system called the endoplasmic reticulum. And there, the ribosomes get together with tRNA and mRNA to make proteins. So this all happens in the endoplasmic reticulum. Protein synthesis does not occur inside the nucleus. The only form of RNA in the nucleus is ribosomal RNA, and then it's there only long enough to get transported out. Okay, so let's go back to one other detail that you need to know, and that is about the centromere. You'll see that at the bottom of the picture, these two red chromosomes, which are really chromatids, and they're joined at some point by a little body called a centromere, and each centromere has a set of proteins called a kinetochore. The kinetochore's function is that's where the mitotic spindle fibers, the microtubules, attach, so that in anaphase, when the spindle fibers start to shorten, they're attached to chromatids and they pull them apart. So that's how the chromosomes get from the center of the cell to the opposite sides. So remember that the centromeres are where the microtubules attach, and they are critical for separating the chromosomes. Okay, so that's a lot on one slide. You'll have to sit when you study and look at this and try to assimilate it all, but be sure you know all the phases and what happens in each one. And remember, this is mitosis. In animal cells, cell division itself, the separation of one cell into two cells is done by a contractile ring, which you see illustrated here. It just pinches the cell in two to make two cells. Well, it's different in plant cells because they have cell walls. So after the nucleus divides and you have the chromosomes at opposite ends of the cell, vesicles containing the precursors of cell wall line up in the middle. Now, these vesicles fuse with each other, making a cell plate, and then there's further fusion of vesicles between the cell plate and the cell wall, so that finally you have two complete daughter cells. So now let's consider the human life cycle. We humans have both a diploid phase and a haploid phase. And that's illustrated in this diagram. The haploid gametes, they have a chromosome number of 23. That's true in both the egg and the sperm. And if the sperm fertilizes the egg, now we have a diploid organism. Diploid chromosome number is 46. And this diploid cell goes ahead and divides and develops into full-blown humans, which have their own sex organs. Humans have testes, males have testes, females have ovaries. So within their sex organs, meiosis occurs, producing the haploid cells again. There's a distinct difference between female haploidy and male haploidy. On the female side, females are born with thousands of eggs. And they're born, so while the female is still a fetus, meiosis occurs and produces egg cells. But they remain quiescent until the female reaches puberty, and then she starts shedding one egg per menstrual cycle. So that's a total of about 500 eggs shed in the course of her life, and that comprises about 42 years. And the female can rely on eggs that she had at birth, or she can produce additional eggs during her reproductive cycle, which is from teenage years through about age 55. And the reproductive phase of females ends with menopause. On the other hand, males are born without sperm. They start making sperm at puberty. Making a sperm cell takes a long time, 2 to 3 months to make one mature sperm cell. But yet, because so many of them are in the process of forming at any one time, millions of sperm are made each day. And in contrast to females, males have a long reproductive phase. There's no, it starts with puberty, and there's no distinct ending point. Some males have a very long reproductive phase, whereas others don't. Okay, any questions about any of this? I'd like to make a point about lifestyles. Different life forms have different evolutionary strategies. I've just been talking about humans. Humans have what's called a diploid dominant life cycle. In fact, most animals have a diploid dominant life cycle, which means that throughout most of their lives, they're diploid. And they're fully formed bodies, they're diploid. And they're haploid only when the testis makes sperm and the ovary makes eggs. But that very quickly after that happens, and that's where meiosis is involved, fertilization can occur, which the sperm fertilizes the egg. And now we're back to diploid. So the haploid phase of humans can be hours or days or weeks, but most of the existence of a human is as a diploid. Now that's in contrast to certain other organisms. And we can take, for example, a fungus. Many fungi, not all, but many have what's called a haploid dominant life cycle. That means they're haploid most of the time that they exist. So the diploid phase can exist for a few hours, but for most of these fungi, if they have any sex at all, they immediately go through meiosis and make haploid cells, which are usually spores. And these spores can go on indefinitely, just propagating themselves by mitosis. Meiosis is not involved. So fungi can be haploid for years, even centuries. And in fact, some fungi have no known sexual phase at all. They live indefinitely as haploids. Okay, so we've talked about mitosis and how it occurs. Now let's consider meiosis, which is a bit more complicated, quite a lot more complicated. The consequences of meiosis: the cell starts as a diploid and it ends as a haploid. The chromosome number is reduced by half from 2n diploid to 1n haploid. And during meiosis, genetic variation is created. Now, this is very important because the genetic variation created by meiosis provides the material for natural selection to operate on and allows organisms to evolve. Okay, there are two cell divisions in meiosis called meiosis 1 and meiosis 2. So if we start on the left here, at the end of interphase, so interphase is the same as it is in mitosis, but then, and the interphase nucleus is diploid. So in this example, in prophase 1, we have 4 chromosomes, which means 8 chromatids. The homologous chromosomes link together. They attach to each other as they condense, forming what are called tetrads. So each pair of homologs is actually double, and so that's 4 chromatids, and that's called a tetrad. And it's while they're in this intimate contact, they can exchange parts. And this is called crossing over or recombination. So this is one of the sources of genetic variation. So this is very important. It happens in every meiotic division. Crossing over occurs. Now, the other source of genetic variation occurs in what's called metaphase 1, where the chromosomes move to this metaphase plate, and they line up there, and the pole, the side of the cell they go to, is entirely random. So any given chromosome can go to one side or can go to the other, and that choice, it's not really a choice, it's just a random event. So that is a source of genetic variation, which side of the cell the chromosome goes to in metaphase 1. Then in anaphase, as in mitosis, the microtubules, microfibers can contract and pull the chromosomes to the opposite sides of the cell. And then the cell divides. So now we're left with haploid cells. We've gone from 4 chromosomes to 8 chromatids per cell, and now we're back to 4 chromatids per cell, 2 chromosomes, 4 chromatids in each of these cells. And this is telophase 1. So this will be a little more meaningful to you after we consider what else happens. Now, 3 sources of genetic variation without mutation. Mutation is a very important source of variation, but there are others. equally important. One is the fertilization step itself. So an egg has not a choice, but it has a probability of being fertilized by any number of sperm. And each sperm is genetically unique. So depending on which sperm does the fertilization determines the genetic composition of the embryo. So that's a source of genetic variation. The other is meiosis. And the two processes are called crossing over or recombination and independent assortment. So let's go to the right here and talk about crossing over first. This occurs only in meiosis 1 prophase. Does not occur in meiosis 2. So you start with a tetrad. Here are 2 homologous chromosomes, a blue one and a red one. And when they form a tetrad, they have points of attachment called chiasma. That's the site of crossing over. So where they touch, in this case at the tips, they exchange tips. So that, if you come down, now one of the blue homologs has red tips, and one of the red homologs has blue tips. So they're now the homologous chromosomes are genetically different from each other. There are different genes on each of these 2 homologs. So when you get gametes, you end up with, where you started out with these 2 blue homolog and a red homolog, you have the 2 parental types, one solid blue and one solid red. Then you have 2 recombinant chromosomes, a blue one with red tips and a red one with blue tips. So that's the result of crossing over or recombination. Now, what about independent assortment? Now this, since there are 2 cell divisions, this can happen in both meiosis 1 and meiosis 2 at metaphase. So there are 2 possibilities. If you start out now, we start with 2 homologs, 2 homologous pairs, blue and red, but the blue ones can be either long or short, and the red ones can be either long or short. So there are 2 different sets of homologous chromosomes. When meiosis 1 occurs, you get this configuration, and then meiosis 2 occurs, so that with this possibility, you get 2 combinations. You get either 2 gametes with entirely blue chromosomes, or 2 gametes with entirely red chromosomes. That's one possibility. The other is shown on the right, whereas where on this side, there is blue and red for the big ones, still blue and red for the big ones. For the short ones, it switched. It's now red and blue. So when the cell divides, you can now get different combinations. You can get a blue long one and a red short one, 2 of those, or a red long one and a blue short one, 2 of those. And this process is entirely random, whether it's possibility 1 or possibility 2. And this gives you a close-up of recombination, crossing over. So we have the homologous chromosomes, red and blue. Each is a pair of chromatids. they come together in very close association, and they can exchange parts. And the simplest way to show that is to show tips. So this random assortment occurs in both meiosis 1 and meiosis 2.
Excuse me, Professor.
Uh-huh.
The previous slide, does that the cross make a problem like a mutation, or it's a
Oh.
normal crossover?
Yes, this is normal. This has nothing to do with mutation.
But there is a change in the series of nucleotide, right? So
Yeah.
when there is a change, it might be a mutation.
It's not a mutation because there's no change in the DNA sequence. It's just that one sequence changes chromosomes. It goes from one chromosome to the other. It's a reciprocal exchange. So where one red chromosome gets a blue tip, a blue chromosome gets a red tip. But there's
So
no
it
change.
doesn't has any effect?
There's no change in DNA sequence on either of those chromosomes. It's just the sequences have changed homologs, gone from one homolog to the other.
Okay, thank you.
But that's an important point, that recombination is not mutation. There's no mutation involved here.
Yeah, thank you.
Okay, now we'll look at meiosis 2. Just to remind you, when we looked at meiosis 1, we ended up with 2 haploid cells. We started with a diploid cell, ended up with 2 haploid cells. That's at the end of one meiotic division. Now we're going to go to meiosis 2. Those same 2 haploid cells are now here in telophase 1, but now they go into meiosis 2, starting with prophase 2, where you have the 2 sets of homologous chromosomes, the red ones and the blue ones, but now recombination has occurred, and so we have both non-recombinant and recombinant chromosomes. Now, when they line up at metaphase 2, again, this is entirely a random process, and either chromosome can go to either pole. And at this point, that the centromeres split so that one chromatid of each homologous pair goes to one side and the other chromatid goes to the other side. Now, I mentioned earlier a term, non-disjunction. That's when chromosomes, when the centromere does not split properly and chromosomes stay together, so that you end up with 3 copies in a diploid cell instead of 2. So that's non-disjunction. It would occur, for example, at this point. All right, so now you have cytokinesis. These 2 cells both divide to make 4 haploid cells. So we've started out at the beginning of meiosis 1 with 1 diploid cell. And now we end up at the end of meiosis 2 with 4 haploid cells. And these are the gametes. These are eggs and sperm. So if you want to look at, just follow, for example, the recombinant chromosomes, they're easier to follow because they have 2 colors. But this blue chromosome on the right here with red tips started out over here. There's a blue chromosome with red tips. And this blue chromosome with red tips went to the pole at the top and ends up here with a small red chromosome. Now we can also look at a red chromosome with blue tips. It ends up in the haploid state with a small blue chromosome. Then you have the non-recombinant chromosomes, a long red one and a short blue one, and a long blue one and a short red one. So you can see that it's a random assortment of the chromosome material. Okay, this is a reiteration of the previous slide. Very important source of genetic variation is independent assortment. You have random alignment of male and female chromosomes at the metaphase plate. So the homologous chromosomes can line up like this, or, so here you have a blue with red tips down. Down here, you have red with blue tips down. So it's just flipped over. So there are two different configurations that have two different consequences. They could line up this way, or they could line up this way, giving a different outcome. And if you follow the chromosomes through, you can see that what you end up with is on the right. You have 8 products of a tetrad. So I've set down some differences between meiosis 1 and meiosis 2. Meiosis 1 starts as a diploid and ends in a haploid, whereas meiosis 2 starts as haploid. So the same haploid cells that are at the end of meiosis 1 are the starting material for meiosis 2. Starts as haploid and ends as haploid. In meiosis 1, we have what's called the reductive division, where one diploid cell becomes 2 haploid cells. Meiosis 2 has what is called an equational division, where these 2 haploid cells from meiosis 1 become 4 haploid cells. In meiosis 1, the homologous pairs of chromosomes separate. In meiosis 2, the individual chromatids separate. In meiosis 1, crossing over occurs, and the result of that, recombination occurs. In meiosis 2, there's no crossing over, no recombination. It's already occurred in meiosis 1. However, random assortment occurs in both meiosis 1 and meiosis 2, and that's because at the metaphase occurs in each of these divisions, and at metaphase, the chromosomes have a random assortment when they decide which pole they're going to. What about the sex chromosomes, X and Y? Well, females have no choice. When they go through meiosis, since they only have X chromosomes in the diploid phase, all they can do is produce haploid eggs. One diploid mother cell produces 4 haploid egg cells. Males, on the other hand, have an X and a Y, so their products of meiosis are 2 X's, 2 X sperms, and 2 Y sperm. But when fertilization occurs, if the sperm contributes an X to the egg, this daughter now has 2 X's and she's a girl. If, on the other hand, and it's equally likely that the father would contribute a Y, when fertilizing, so that the cell, the zygote that's formed, is XY, and that will produce a son. So this is, again, a reiteration, but it's a side-by-side comparison between what happens in mitosis and what happens in meiosis. These processes have two completely different goals. Mitosis, the goal is to make two identical cells from one. So this is just a repeat of what we've gone over before. You have prophase, metaphase, and you get two diploid cells. You start with diploid, you end with two identical diploid cells. The daughters are identical to the parent. In the case of meiosis, you have two divisions, meiosis 1 and meiosis 2. And as a result of the recombination events that occur in both meiosis, well, the recombination in meiosis 1 and random assortment in both meiosis 1 and meiosis 2, you end up with daughter cells, 4 haploid cells, none of which is identical to the progenitor. So this is where you can contrast the two processes. The goal of mitosis is to give identical products. The goal of meiosis is to give non-identical. Every product of meiosis is different from the progenitor cell because of the two genetic variation processes that I've mentioned over and over. Why is meiosis important in evolution? Well, evolution requires variation, and meiosis is an endless source of variation. It never stops. With every meiotic division, more variation is produced. So natural selection can operate on all this variation and sort out the most fit genotypes. Which set of genes is best adapted to the environment at that point in time. And to reiterate, variation can arise from crossing over. That's recombination, and that happens in meiosis 1 only. Variation can arise from random assortment, which occurs in both 1 and 2. But why is meiosis 2? Why do we need 2 meiotic divisions? Why is meiosis 2 necessary? Well, if cell division stopped after meiosis 1, each sexual generation would double the chromosome number. And as generations went on, the number of chromosomes would become immense, and that would be lethal. That's why we have the equational division in meiosis 2, to ensure that the organism's ploidy level after fertilization remains as a stable diploid. So this is both a summary and an indication of why meiosis is so important in the evolution of higher organisms. And note that mitosis involves neither crossing over nor random assortment. Mitosis has a completely different goal. It wants to produce identical progeny, whereas meiosis wants them to be different from the progenitor. Now I've put a little table here at the end, things that I'd like you to think about when you study. This is sort of a semi-study guide that I won't go over here. You can think about these things as you study this material. But these points are to try to guide your thinking to some of the most critical parts of this lecture. And that's the end for today. Any questions or comments before you go off to do quiz 6?
Yes, Professor. This is Kat.
Uh-huh.
Why do parents have two different children? One children with syndrome and the other children is normal?
Okay, one child is normal and the other is abnormal?
Yes, or syndrome.
And you're saying, why does that happen?
Yeah. With the same parents.
Okay, the same parents can have a normal child and an abnormal child.
Yeah.
Well, just take, for example, supposing one of the children inherited a cystic fibrosis mutation, that would cause that child to be different from the other. Is that what you mean?
Yeah.
So a mutation can make the difference. But different children, I mean, no, unless they're twins, no two children are alike, and that's because of genetic recombination and random assortment. Different children can have the same parents, but they get different combinations of genes.
Professor, I have a question. Oh, sorry.
Oh, go ahead.
Okay. You were talking earlier about like fungi and how they do meiosis as well. Do we classify? Do they have sex cells like X and Y like humans do? Like, do we classify them male and female?
No, they don't have sex cells like animals do. What they have is mating types. So mating types are just two different genetic sequences that, on normal chromosomes. They're not sex chromosomes. They're just two different genetic sequences that have to pair with the opposite sequence in order to have meiosis, in order to make a zygote, and a zygote, and then go through meiosis.
Okay.
So
Thank you.
the answer to your question is fungi have sex, but they don't have sex chromosomes.
Okay. Thank you.
All right, this is Zach. I had a question about I think it's anaphase. I think. So could we think of the kinetochores as sort of like two very strong magnets put together, and the microtubules as sort of like winches trying to pull those two apart to the different sides of the cell before it splits?
Yeah.
It's a weird sort of analogy, but like this is kind of like how I
Yeah.
try to bring it to like a physical analogy.
And I think that's an accurate analogy. The microfibers, microtubules, are attached to the kinetochores on the centromeres. And when the centromere splits, it releases the two chromatids from each other. So they're no longer tied together. And then the microfibers, they're condensing, they're contracting and pulling the chromatids away from each other to opposite poles. So yeah, you could think of it as, think of the centromere as a magnet that gets pulled away from its opposite pole.
All right, cool, thanks.
I was wondering about like trisomies. Are there like certain factors that lend themselves to like creating those, or?
Well, as I understand it, there's no systematic mechanism behind trisomies. It's just that mistakes are made. Just like copying errors are made when replicating DNA. When chromosomes separate, they can make a mistake. And instead of separating, two chromatids can stay together. If they do, now you have a sex chromosome with a a sex cell with a double chromosome. And when fertilization occurs, now instead of 1 and 1 is 2, we have 1 and 2 is 3. And that can occur with any chromosome at sort of it's a random frequency.
Thank you.
So we rarely see trisomies because they're lethal. Most chromosomes, if they're there in 3 copies, the fetus dies before it's ever born. And I pointed out the ones that can live. Okay, anything else? All right, then I will stop sharing.